Good afternoon. My name is Laura van Moortel and I am a shared PhD student between the groups of Professor Caroline de Bosser and Professor Chris Gevaert in the VIB Center for Medical Biotechnology in Ghent, Belgium. I would like to thank the organizers for the opportunity to present my work today, which is focused on the glucocorticoid receptor and more specifically on the search for improved ligands for it. So, as you may or may not know, glucocorticoids are still among the most frequently prescribed anti-inflammatory agents in the clinic. So they are used for the treatment of inflammatory disorders, also for hematological cancers, and they're also prescribed to prevent organ rejection. Unfortunately, even though they are very cheap, they come with a very long list of side effects, which is, of course, a very big drawback and a reason why some people don't want to take glucocorticoids. So these side effects include hyperglycemia, osteoporosis, muscle wasting, mood disorders, and so on. It's a long list. And on top of that, a substantial fraction of the patients develop therapy resistance over time. So that's the reason why people have been looking for improved ligands for the glucocorticoid receptor for quite some time now. And before I'm going to explain to you what exactly we mean with these improved ligands, I'm just going to give you a very brief introduction on the biology of the glucocorticoid receptor. So GR is part of the nuclear receptor superfamily, and this protein is mainly located in the cytoplasm in absence of ligands. And then once it is bound by glucocorticoids, it translocates to the nucleus, and there it acts as a transcription factor. So its actions and its signaling mechanisms are very diverse and are also very cell and gene specific. But for the clarity of this talk, I'm going to focus on two important mechanisms here. So GR can either form homodimers and binds directly to so-called glucocorticoid response elements or GREs, and this leads to the upregulation of downstream target genes. And while this mechanism also contributes to anti-inflammatory effects, it is mainly linked to a lot of the metabolic side effects of glucocorticoids because it leads to the upregulation of a lot of genes that are involved in this mechanism. For instance, PepsiK, glucose 6-phosphatase, and TAT, which all contribute to hyperglycemia. On the other hand, GR can also stay as a monomer and interfere with the activity of pro-inflammatory transcription factors such as NF-kappa B and AP1, and this then indirectly leads to the downregulation of pro-inflammatory cytokines. So in the past, it was always hypothesized that GR exerts this interference with NF-kappa B and AP1 activity via protein-protein interactions. But more recently, it has been shown that this protein can also bind directly to NF-kappa B and AP1 response elements in the DNA. So the main message from this slide is that the monomer-driven effects of GR are mainly linked to its anti-inflammatory effects, while the dimer-driven effects of GR are mainly linked to the side effects. So that's why people were interested in the development of so-called GR modulators, and more specifically in the development of semograms, which are selectively monomerizing GR ligands. And the idea is that these ligands would would cause less GR dimerization and therefore less signaling via GRE-driven mechanisms, which would then lead to a reduced side effect burden. But on the other hand, it would still maintain the monomer-driven effects of GR, so these compounds would still lead to the strong anti-inflammatory effects that we also see with the classic full agonist glucocorticoids. There has also been some interest in the development of C-digrams, which are then compounds that signal more via dimeric GR, because it has been shown that these dimer-driven effects are very important for combating very acute inflammations, such as, for instance, sepsis. So that's why people were also interested in C-digrams. But then more specifically about the semograms, there has been a lot of research on it, and yeah, a number of compounds have progressed to clinical trials, but unfortunately they all failed at some point. So for now there are still no semograms in clinic. The reasons why all these compounds failed are, yeah, of course, very diverse. And if you're interested in reading more about it, I could recommend the Perspective article that we published about this in Frontiers in Endocrinology in 2020. But today I would like to focus on one potential contributing factor for all these failed compounds, and that is that the current screening assays that we use to characterize novel glucocorticoids, that these might be oversimplistic and, yeah, ignore the very complex nature of glucocorticoid signaling. So that's why we decided to yeah, obtain some powder stocks for a number of different GR ligands. So we have a number of classic glucocorticoids that are still used in the clinic. And then we were also able to obtain some powder stocks for many of the semograms that were published over the years, or at least compounds that were claimed to be selective GR modulators. And we wanted to test these in the current screening assays and then compare their activity profiles with their outcomes on endogenous GR target genes to see to which extent yeah, these effects would actually correlate with one another. So first we started with the classic screening assays, which are typically luciferase assays. Usually it's a GRE coupled to luciferase to monitor the dimer-driven effects of GR, so to monitor the side effects. And then on the other hand, we also had an luciferase reporter that was driven by an NF-kappa B response element to monitor the anti-inflammatory effects of glucocorticoids. So we started by characterizing the full agonists that we had in these two screening assays. So both reporters were stably integrated in A549 cells. And here you can nicely see the dose response curves for the agonists in both of these assays. And so here the NF-kappa B reporter, yeah, the signal goes down because first we need to trigger or stimulate inflammation with DNF and then suppress this inflammation again with the compounds. So you can nicely see that this worked. And you can also see that the antagonists, they did not really have an effect on either of these two reporters, which was also what we expected. So then we also screened the other ligands, so the segrams that we obtained in these reporter gene assays, and based on their activity on these two reporters, we divided them into different classes. So here I'm showing you the data also for the semograms and the CAD-grams. So here you can nicely see that the semograms have yeah, a lot less activity on the GRE-driven luciferase reporter. It's less than 50% of dexamethasone, which we took along as the gold standard. And on the other hand, you can see that these compounds still had strong anti-inflammatory effects on NF-kappa B. And then the CD-grams, on the other hand, these had stronger effects than dexamethasone on the GRE-driven luciferase reporter, which is, yeah, what we wanted to see because that's why they are defined as CD-grams. And they also still had very strong anti-inflammatory effects. And then we also had some compounds that had kind of an intermediate signaling profile, and those we classified as partial agonists. So then we selected a number of compounds from these 5 groups. So yeah, these 4 and the partial agonists. And then we went forward with these compounds and we used them to treat A549 cells. And then we looked at the mRNA levels of a number of well-known GR targets. So here you can see the outcomes on the GRE-driven target genes. So while the activity profiles of the compounds in general were quite comparable to what we saw based on the luciferase assays, there was one thing that was very clear, and that is that we were not really able to recapitulate the CD-gram effect, because here on the qPCR we did not see any significant differences between the CD-gram compounds and the dexamethasone control. Only for glutamine synthetase and the condition without TNF, which is the open bar, there was a significant difference, but it was the only condition where this was the case. On the other hand, we could still see that the semograms had less effect on the GRE-driven reporter, which is nice because this still shows that they could lead to less therapy-associated side effects. But unfortunately, on the other hand, we noticed that they did not have the same anti-inflammatory effects as, for instance, dexamethasone, which was a bit unfortunate. And then next, we also wanted to study the effect of these compounds in another cellular model because we know that the effects of glucocorticoids can be very cell type specific. And therefore, we also wanted to test these compounds in a cell model that was more relevant for the metabolic side effects. So that's why we also tested them in hepatocytes. And while we saw that for some genes, the effects of the compounds between the primary hepatocytes and the A549 cells were very similar. For some other genes, we also saw that there were quite some differences between the two cell lines. So this also shows that it's very important to screen novel glucocorticoid ligands in cell models that are relevant for the disease you want to study later on, and also for the potential side effects that you want to study. So the main conclusion from these data so far is that the luciferase reporters seem to be more or less okay, but they appear not to always be the best predictor for the compound effects on the endogenous level. So that's why we decided to develop 3 novel assays that are also suitable for high throughput screening. And we were hoping that these assays would have an improved predictive power for the effects on the endogenous. So first of all, we introduced the Nanobit system to monitor GR dimerization. So the concept of Nanobit is that you couple, in this case, GR to 2 portions of the nanoluciferase, a small bit and a large bit, which have very low affinity for each other. So only when you add a glucocorticoid that then causes GR dimerization, you will get reconstitution of the nanoluciferase. So in this manner, you're able to quantify GR dimerization based on the luciferase signal. And here you can see nicely that the semograms had, or most of the semograms had significantly less dimerization than dexamethasone. But on the other hand, we again saw that the CD-grams, they did not really show any difference with dexamethasone and the other classic agonists, which was a bit puzzling because we expected that these compounds would have higher activity here, since they are supposed to be more active on GRE-driven signaling mechanisms. Then the second assay was a way to monitor GR protein levels, because we know that the downregulation of GR protein levels is one of the mechanisms contributing to therapy resistance. So that's why we used a CRISPR-Cas knock-in system to introduce a HYBID tag at the N-terminus of GR, also in A509 cells. And this HYBID tag is also a portion of the nanoluciferase, but this time it has very high affinity for the large bit. So if you treat your cells with glucocorticoids and then afterwards you add your large bit and your substrate to your lysates, you will get reconstitution of the nanoluciferase, and the signal will be in direct relation to the amount of GR that was present in your cells. So here you can see that this assay also worked nicely, and that you actually saw downregulation of GR levels with all the compounds that we studied. But we could still see that there was a significant difference between the semograms and dexamethasone, which shows that these semograms also might cause less therapy resistance than classic glucocorticoids. And again, we saw that the CD-grams did not really show any differences with the classic agonists. Third assay was also based on the hybrid knock-in in A509 cells. And here we applied it to quantify GR serine 211 phosphorylation, because we know that this is a very important hallmark for GR activity, especially via GRE-driven mechanisms. So here we added an antibody against this particular phosphorylation site, and then a fluorescent secondary antibody. And then you get a breath signal, and in this way you are able to quantify the occurrence of this post-translational modification. And here we again saw the same trends as with the previous assays. So there was a significant difference between dexamethasone and the simogram compounds, which is again in line with their reduced activity on the GRE-driven mechanisms. But here we again saw that there was not really a difference between dexamethasone and the siligram compounds. And then in the end, we wanted to find out which of the luciferase-based assays was actually the best predictor for the outcomes on endogenous target genes, so the genes that we evaluated via. And here we saw that for almost all the genes that we evaluated via qPCR, that the breath assay monitoring GR phosphorylation was actually had the highest predictive power. So this shows that the novel assays that we developed, that they indeed might have higher predictive power, and that they might indeed be an improvement over the already existing classic screening assays. With that, I would like to conclude by thanking all my colleagues from the lab of Professor Caroline de Bosser, and also my colleagues from the group of my co-promoter Chris Gevaert. And then I would like to thank you for your attention, and I'm happy to answer any questions.